All right, I want to thank you all for coming. I'm very excited to be doing this. This spotlight session is on shapeshifting and the control of protein function. And we've lined up a series of speakers who are going to be talking about different aspects of protein function, many of them related to quaternary structure. I'm going to give a presentation pretty much describing why I find this particular topic so fascinating. Nancy's going to be talking about shape-shifting as it refers to, I guess, protein nucleic acid interactions. Carrie's going to be talking about it as it relates to circadian rhythms. Jeff is probably going to be focused on a lot of the kinetic phenomena that occur when you have some of these kinds of shape-shifting. Connie's going to talk about moonlighting. And finally, Sarah's going to talk about a wonderful transforming viral protein that can undergo many shape changes. Okay, I wanted to start by talking about morpheeins. These are multimeric proteins that can come apart, they can change shape, and they can reassemble differently with functional consequences. And the story starts about 20 years ago with the unexpected behavior of a metabolic enzyme that we had been studying for close to 20 years. In this case, there are two assemblies, an octameric assembly and a hexameric assembly. And the interconversion was shown to require a conformational change in the dimeric form. In other words, this protein had to come apart, change shape, and come back together again. And the rate determining step here was shown to be a hinge motion between two folded domains. Now, it turns out it happened at the level of the dimer. So you'll notice that the classic idea of refolding isn't implied here. For instance, parameters like intrinsic fluorescence or circular dichroism didn't discriminate between these alternate assemblies. It was just a hinge motion. The other interesting characteristic is that the dimer wasn't easy to see. It was at 0.2%. So oftentimes when I talked about this, and I said, well, it has to go through a dimer, maybe your protein is the same, I would hear things like, well, I don't see a dimer, I don't see a smaller multimer. Well, we didn't see this either. We had to look really hard. Now, there are two levels of physiologic relevance for this particular protein. One is it's responsible for the allosteric regulation of enzyme activity. It turns out only the octamer has proper subunit-subunit interactions to gate access to the active site. And second is that perturbation of this equilibrium was shown to be the structural basis for an inborn error of metabolism. So this wasn't a laboratory artifact. Furthermore, most importantly, the position of the equilibrium can be modulated by ligands binding at what I like to call assembly-specific binding sites. And I think this is going to be a theme that we hear again and again today. We realized that this was sort of a different mode of allostery. It wasn't Monod-Weyman-Changeau, and it wasn't the Koshland. It wasn't necessarily concerted or stepwise. So we called them morpheeins. Now, I've learned that inventing a word is not necessarily a great thing. However, it's also importantly a general mechanism for the design or action of biological modulators, such as drugs. So here we have a new approach to drug discovery. And most interestingly, because this has to do with the coming apart and going back together again of multimeric proteins, the equilibrium responds to pH. And here I'm going to show you my one data slide. Oh, I'm sorry. Here's the allosteric ligand. Let me see if I can find here's the allosteric ligand binding site. It's forming, its position is a subunit interface that is present in the octamer, but not present in the hexamer. Okay, this is my data slide. Now it turns out for this protein, we had the isolated wild type protein, and we also had a stable single amino acid variant. And if you look at the pH rate profile, you'll see that for the wild type protein, you have a standard bell-shaped curve. And for the hexameric protein, you have a different looking curve. And if you do some kinetics on it, you see that the Km here is low and the Vmax is high. The Km here is high and the Vmax is low. But if you go to pH 9, what you see is that the Km for both of them is the same. And in fact, if you vary the substrate concentration over a very wide range, you'll see that there are two Kms. There's the one for the very small amount of the octameric component, and then one for the hexameric component. So it turns out for this protein, doing assays at 2 different pHs became a diagnostic for the quaternary structure. But the point I want to make is that this arm of the pH rate profile isn't deprotonating something at the enzyme active site. It's the transition from one assembly to a different assembly. Okay, porphobilinogen synthase is the enzyme I've been telling you about. It is a fascinating enzyme. I could talk about it for hours. I'm not going to. We put a paper out a few years ago. If you're interested in this system, I recommend that you look at this. But what we really wanted to ask was, okay, we have this peculiar behavior for porphobilinogen synthase. What might be hints of other multi-domain multimeric proteins that share this dissociative allosteric mechanism? We realized that this one sequence, one structure, one function paradigm had caused us to have difficulty to see this phenomenon. How much has that masked recognition? We saw that chemically equivalent isoforms, the octamer and the hexamer, were separable by ion exchange chromatography. How many reports might there have been of this sort of thing in the literature? We saw single amino acid substitutions that caused a gross disproportionation of this equilibrium. What we had called years ago long distance effects, and maybe they're still called that. How many examples are there of crystal structures, of constructs that clashed with each other? And here the example is HIV integrase. It's a 3-domain protomer. If you get a crystal structure of domain 1 and domain 2, and you superpose it on a crystal structure of domain 2 and domain 3, domains 1 and 3 clash with each other. This is often thought of as a crystallographic artifact, but maybe in fact it's a hint that a protein can have 2 different conformations, where the conformational change is a hinge between 2 folded domains. And finally, there is that double hyperbolic kinetics, which is what Erwin Siegel had written about in his classic text as a mixture of isozymes. You have one protein, but one assembly has a high Km and a low Vmax, and the other assembly has a low Km and a high Vmax. And so you see this double hyperbolic kinetics. We also came to appreciate that we might never have seen this hexamer if certain things hadn't been in place. For one thing, if we were isolating our protein using purification tags, we never would have seen these things separating on an ion exchange column. If we had continued to follow the old rule of only collecting fractions of the highest specific activity and tossed everything else, we'd have never have seen the hexamer. In fact, we'd been tossing the hexamer for more than 15 years. Or what if we hadn't just happened to study a particular disease-associated variant that fortuitously formed a very stable hexamer? And you'll see some examples during today's talks of our investigators studying particular variants that were either designed or found to stabilize a particular multimer, or for that matter, if we didn't happen to get the crystal structure for the hexamer stabilizing variant. So I realized that an awful lot of sequence and structure-based predictions are what drives much biomedical research. And the question immediately comes up, how might we have predicted this morpheein phenomena, proteins that can come apart, change shape, and go back together differently? So in the mid-2000s, we put out a paper in Tibbs calling the morpheeins and talking about it as a structural paradigm for allosteric regulation. This is a rather complicated slide of different multimers, but the point is that each multimer is both structurally and functionally distinct, and that there can be multimer-specific ligands that act as allosteric regulators. Now, this paper also predicted the morpheein character of a number of proteins. One of them is ribonucleotide reductase. And for that, I ask you to look at the current work of Nozomi Ando or Iman Ai, who are both working on this fascinating aspect of ribonucleotide reductase. But finally, in 2008, we had used computational docking to find something that bound to the hexamer and would act as an inhibitor. And when we did this, we were asked to make a cover slide for a cover photograph for the cover of the journal. And that's when we came up with this model, which I think is much nicer to look at. So here we have two different die shapes, a cube and a pyramid. And if we say that these are in equilibrium with each other, you can think about it as phase 5 and phase 6 coming together with each other. If we think about this and we say, oh, there's a rule for assembly that phase 1 and phase 4 come together, then this cubic die is going to assemble as this tetramer, and the pyramidal die is going to assemble as this pentamer that looks like a flying saucer. So you can imagine how the function of these two things might be the same, how the interaction partners of these two things might be the same. But most importantly, what you see is that this particular assembly has a multimer-specific surface cavity that can act to bind drugs or allosteric regulators. Okay, so what is a morpheein? These are homomultimeric proteins that can come apart, change shape, and reassemble differently with functional consequences. Is this the 5th or 6th time I've said that? The stoichiometry of the assemblies need not be different from each other as they are in PBGS. For that matter, they may not be finite. Yesterday, we heard a lot about filament formation. And it is true that the structure of the individual components of a filament may be the same in the filament and not in the filament, or they may be different because they had to undergo a conformational change. The functional distinction could be like our PBGS system, on versus off, or they could be moonlighting functions, and Connie's going to talk about moonlighting. The equilibrium position can be modulated through binding assembly-specific ligands. And I think nearly every talk we're going to hear today is going to talk about that aspect. The equilibrium position responds to single amino acid substitutions, sometimes really dramatically. And the interchange of assemblies can be quite slow, which again, I'm not going to talk about. Well, this languished for a while until 2013, when the Ullman Sapphire lab finally showed another protein that behaves in this way, and this is the VP40 protein of the Ebola virus. In the VP40 protein of the Ebola virus, there are at least 3 different assemblies with 3 different functions, and we'll hear a little bit more about this later today. But you note that the example here shows that the conformation of the monomer within a given assembly may not be uniform throughout the assembly. So that's even a further level of complexity than what we had seen. So in 2015, Erica and I and my then student, Emily Arturo, who's currently working with Erica, got together to talk about how the community of protein scientists resisted the idea that proteins could have more than one shape, more than one fold in a physiologically relevant interconversion. But the cool thing that came out of this little manifesto is that we lack the rules to predict these. And I think that's the big unanswered question right now. So what are the opportunities to this community? Well, we could compile and test a list of proteins that might be morpheeins. And in fact, to that event, we made a Wikipedia page. Trevor Selwood did a huge survey of the literature, most of which was done manually because these things often don't show up in abstracts. We actually have currently picked one phenylalanine hydroxylase, which is the protein my laboratory is currently working on. So the other opportunities are focusing on how to predict what proteins might be morpheeins. For example, we might ask about the moonlighting proteins. And the last one is to promote a community of investigators, like the people here at this webinar, who are looking at various aspects of protein shapeshifters and bring these people together as a community. So, to that effect, we had a session at the ACA meeting in 2018. We have this spotlight session, and I ask you all to look at the 2021 Protein Society meeting, which is currently scheduled to take place in Boston, where we will again focus on this quaternary structure shapeshifting as a way to promote alternate functions. So just some acknowledgements. These are the people in my laboratory who had done a lot of the work over many years on the PBGS morpheein system, which we haven't worked on in a while. Most recently, Emily Arturo. This is the work that got Roland and I working together. Roland has spent a lot of time focusing on quaternary structure and how that information comes out of the PDB. We've had a lot of good financial support, and mostly we've had excellent support from the Fox Chase Cancer Center. So with that, I have gone over my time, and we will move to questions.
Okay, yeah, that was great. We do have a question, so you have about 2 minutes. So the question is from Anjana George. Do you think this is possible in the case of membrane proteins?
Yes, yes, I do, except the rules would be different. Because in the case of, for instance, PBGS, the interface that comes apart and goes back together again in the crystal structure has a lot of ordered water molecules. So it would be just the opposite in a membrane protein, where you would have to have a hydrophobic interface that then comes apart and goes back together again. But yes, absolutely.
That's really interesting. Okay, another question from D. Arsenan Green. Does changing the pH change the relative amounts of octamer versus hexamer, or just activity?
It changes the relative amount of octamer versus hexamer, and of course I couldn't show you that, but you can see it using dynamic light scattering, which is a very nice way to watch that transition.
Is this activity, it's a way to regulate the enzyme in vivo, so is the inactive oligomer like a storage state for the enzyme? Or
that's a fascinating question. And in fact, I believe in plants, that is what happens. So this enzyme, I didn't talk about its function at all. It catalyzes the first common step in the biosynthesis of all the tetrapearls. And so it's needed for chlorophyll biosynthesis. And of course, if you start making chlorophyll precursors in the dark and the sun comes out, those precursors are photolabile and your plants will burn up. In fact, the substrate for this enzyme was investigated as a herbicide in the mid-1980s. You spray it on the field, makes intermediates, sun comes up, the weeds die. It was good. Not a very stable molecule, unfortunately. So one of the very first things in the light reaction, when light hits the plant, is that magnesium floods into the chloroplast, and the allosteric activator in the plant enzyme is magnesium. So this is one of the ways that photosynthesis is turned on.
Cool, great. So I think we're over time now. So there's still some questions in the question and answer box, but I guess we're moving on to me.
Yes, we are. So then I would like to introduce Nancy Horton, who's going to be telling us about protein nucleic acid interactions as they relate to this multimerization and filamentation.
Thank you. All right, thank you. I think I'm on the wrong setting. Multitasking. Okay, so yeah, thank you. Thank you for the invitation. It's great to be here and be part of this group. I'm going to be talking about our work on a nuclease called SgrAI1. As you may have seen yesterday in the webinar, I talked about it in the filament forming enzyme session. It does form filaments that are activated in their DNA cleavage properties. And also interestingly, they change their DNA sequence specificity when in the filaments. So yesterday I talked about a kinetic investigation, and that webinar should be posted soon. somewhere near you. Today I wanted to talk about a structural investigation. So the questions that we're looking at are, one, how does filament formation activate enzyme activity? And two, how does filament formation expand the specificity, the sequence specificity? So what I mean by that is, well, the fact that this is a type 2 restriction endonuclease from Streptomyces griseus. It recognizes this 8-base pair recognition site in DNA, duplex DNA includes here. If there's only a single copy of this recognition site in the plasmid, it'll bind it tightly, but cleave it only very slowly. If there's more than one copy, it cleaves them all very rapidly. This is a really interesting part. So it will also start cleaving other sequences we call secondary sites. They differ from the primary in the identity of the outer base pair or the second base pair. The enzyme binds tightly to them, nanomolar affinity, but it doesn't cleave them unless there's also a primary site, the primary site DNA sequence in the same DNA. Then it will cleave all of these sites rapidly. So this primary site sequence seems to be modulating the activity of the enzyme. It activates it for DNA cleavage, and it alters its DNA sequence specificity to cleave 14 additional sites. After many years of biochemical and biophysical investigations, we came up with the following model. We learned that our enzyme forms filaments when it binds to the primary site. And in this filamentous form, it's activated in DNA cleavage. So there's something different about maybe the enzyme structure that activates its DNA cleavage properties. But when it's bound to the secondary site, which differs from the primary in only one base pair, it's not prone to form these filaments for some reason. But if you have a mixture of the two, the primary site-bound enzyme will drive filament formation and drag in enzymes bound to the secondary site, thereby activating them for DNA cleavage. So we wanted to understand structural aspects of this mechanism, and we determined crystal structures. I'm not a crystallographer, so that's what my lab does. And it looks a lot like other type 2 restriction endonucleases. It's a homodimer down to one duplex of DNA. We solved the structure with the primary site and the secondary site, and they look virtually identical. Each subunit has an active site that cleaves one strand of DNA. It's a magnesium-dependent enzyme. We do see two divalent cations binding in the active site, but one of the two is displaced from where we think it should be, and we think that's the origin of the low DNA cleavage activity in this conformation. So we also determined the structure of the filamentous form, this time in collaboration to determine it by cryo-electron microscopy. And we found that it's a left-handed helix with about 4 copies of the enzyme bound to DNA per turn. The filaments can get quite large, up to 30 or more, and presumably it can grow infinitely on either end. But in at least the experimental setting, we're seeing filaments 30 or shorter. There is a change in conformation of the enzyme. So here in this movie, I'm morphing between the two conformations that found in the crystal structures of the low activity conformation and that found in the filament with the activated conformation. And these conformational changes, we think, are made to maximize interactions with neighbors in the filament. Next one. Okay, so globally, this conformational change is basically a rotation of one subunit relative to the other by about 9 degrees. To accommodate that rotation, there are also changes in position of residues at the dimeric interface. And these residues lead directly into the active site, which we think impacts the activity of the enzyme. So to orient you, this is our cartoon of DNA. This is the bond to be cleaved right here. Metal ion A positioned here. It binds the nucleophile, water, hydroxide, for nucleophilic attack. And there should be a second metal ion according to the 2 metal ion mechanism. But we don't see that in our crystal structures in the low activity state. The metal's there, but it's further away, very far away. So we think that's why DNA is cleaved only slowly in this conformation. But in the filament, we see that change in conformation, and that brings in a segment that has a carbonyl that's now positioned very nicely to bind a second metal ion, we think. Unfortunately, we didn't have the phosphate in our EM structure, So we don't actually see metal B, but this is a hypothesis we're now testing with additional structures. Okay, to kind of tie it together, we see the rotation of one subunit to the other when we morph between the low and high activity conformations. And we'll zoom into one of the active sites to look closely at the metal ion position. So you see how a change at the dimeric interface affects the segment, moving a carbonyl closer, and we think to ligate the second metal, which should now provide rapid DNA cleavage properties. Okay, so that's how we think the enzyme's activated when it's in the filamentous form. But what about this business of changing it or expanding its sequence specificity? So we had our model here about forming filaments, but in this case, we expand it to include conformational state. So in the low activity conformation, we call the T state. This has the weak metal binding site. It's low DNA cleavage kinetics. We think it's an equilibrium when the enzyme's bound to DNA with the other conformation, the high activity conformation. Of course, when it's in, cleaving DNA slowly, it's mostly in the T state. When it occupies the R state, when it flickers into that, it has the opportunity to now assemble into a filament with activated DNA cleavage because we have that change in conformation that brings in the second metal binding site. So really, to determine how fast you cleave DNA, you need to adjust this conformational equilibrium, just like Eileen was talking about. So if you have an enzyme that's always in the T state, it'll never access the R state, it won't form filaments, it won't cleave DNA. If it's always in the R state, there's no allosteric. It's always going to form filaments and always have activated DNA cleavage properties. But there's an equilibrium here. It favors the T state in order for this to be allosteric. Both states, the T and the R, are favorable. This is our hypothetical free energy diagram, but it's more favorable in the T state. So when we move from the T to the R, it's an uphill sort of thing. And again, the R state, though it's less stable than the T, overall the system has more energy when it forms the filament because of those contacts with the neighboring copies in the filament. So we think there's maybe a perturbation of this equilibrium due to the substitution in the secondary site relative to the primary. So one base pair difference must be enough to perturb the energetics, to perturb this equilibrium, to favor the T state a little more. So that could be by preferentially stabilizing the T state. Maybe there's some interaction with the DNA of that base pair that prefers the T state, or it may just destabilize the R state. Maybe there's something that happens in the R state conformation that is less consistent with this particular DNA sequence. So overall, when you go from T to R, it's uphill, and you have less filamentation. It's not so bad that you can't ever go into the filaments. It's just enough that we don't see it, at least under our experimental conditions. So we wanted to know can we look at the structures and maybe understand what's giving rise to this perturbation of these energies? These are the structures we have. We have the crystal structure of the enzyme with the primary site sequence and also with one of the two secondary site sequences. And these are pretty much identical. We have one cryo-EM structure at 3.5 angstrom resolution, which is actually pretty good for EM. We don't have the structure with the secondary site, only the primary. So really to understand this whole kind of diagram, energy diagram, we need all four structures, but we only have these two. So we'll compare these two and look to see if we see any differences in the conformation that might relate to energies when we substitute the base pairs. So we looked for protein DNA interactions that may be different in the T and R state, particularly at these base pairs that are substituted in the secondary site, but we didn't see anything like that. The only thing we saw was a difference in the DNA conformation between the T and R state. So it could be that this change in conformation, maybe the secondary sequence, but that particular DNA conformation favors the T and disfavors the R. That's the idea. So here we're highlighting it for the two structures. We superimpose the CG base pair and then look where the AT is, and we see that it's shifted. Here's just the morph between the two, and you see how it affects the stacking. Now, stacking is one of the main contributors to the stability of DNA duplex structure. And so it's very important contributor to the overall energy. So in this diagram, we measured the stacking area from the structures. And we actually see there's more stacking energy in the R state. So it's, usually stacking area follows energy. So that means going from the T to the R is actually gaining energy. So strangely, even though going T to R is an uphill battle overall, it's actually favorable for this particular change in conformation. And if we look at the primary site sequence, we can see there's a pretty good gain in energy when we go from the T to R, increasing the stacking area. Now, what about secondary site sequences? Well, yeah, there's a gain in stacking energy, but not nearly as much. So what we think is this perturbs the T versus R energy, the ratio between the two energies, because the primary site is contributing more energy to the R state than the secondary site, just merely because of this DNA structure. This is our hypothesis that we're now testing. This is a form of indirect readout of DNA sequence because we're using the sequence-dependent DNA structure and energy to differentiate between different states. Okay, that's my talk, and I'd like to thank people, the graduate students and undergrads who did this work. These are EM collaborators, very talented electron microscopists. Dmitry Lemkus has been our ongoing collaborator at the Salk Institute and funding. And thank you. I'll take your questions now.
Thank you, Nancy. I'm looking for open questions. I am not seeing any open questions. You might have gotten all your questions yesterday.
Yeah, I talked about the kinetic structure. I stumped them.
Maybe
Well,
so.
It's very morphine-like in the sense that we have this equilibrium between the two conformations, one conformation being more active and the other not. We don't have like that fourth conformation of, the oligomer that's inactive.
There's a question. And the question is, how was the DNA sequence specificity of the enzyme originally characterized?
So my collaborators were also at New England Biolabs, looked at this first, and they, usually purify these enzymes and then react them with lambda phage, and then they determine the sites that way. And so they did early on see this weird activity. It was shown to be, at first they thought it was forming kind of like a tetramer, two dimers coming together. There's restriction enzymes that do that. And so they're really concerted and cooperative. They need two sites in order to cut. But this one was a little different, it's behaving differently. And so that's where my lab stepped in to really try to understand it. I was intrigued by this change in DNA sequence specificity that I wanted to understand.
All right, there's one other question, and that is, are there other restriction enzymes that act similarly, or is this rare?
This is the only one we found, but even though people say, oh, everything's known about restriction endonucleases, it's not true. There's like 3,000 of them. How many have been characterized? Only a few hundred. So there's probably a lot more interesting stuff out there. Remember, this is a highly evolved system, since the beginning of life on Earth. We've had early forms of life, bacteria and phage. It's one of the highest biomasses on the earth, right? In the ocean and the soil and even in your gut, and a very fast life cycle. So there's a high turnover of, these kind of like, it's like a war, right? The phage versus bacteria, attack and counterattack going on, for so it's an evolved system.
I just wanted to comment. I think here you have an example of, you don't see it if you're not looking for it.
Exactly, because people thought this thing, it's like really poorly behaved, it keeps precipitating. In fact, I had a lot of resistance because people kept telling me it's just precipitating. And I'm like, really? Because it's more active as the precipitate. So you had to look more closely. There's a lot, you talk about resistance, a lot of resistance. People told me, enzymes don't work this way. And that's why I started looking at all the ones that could form filaments, just like looking at all the ones that form morpheeins.
Yeah, I think we all share that. This was part of that dark corner in the bar in Boston, was Erica and I going, there is so much pushback against this because people keep saying enzymes don't work this way.
Exactly. Yeah.
There was less pushback because hers was a viral system, as if virus proteins are special.
They're allowed
proteins are proteins.
Yes.
Viruses may be special, but I don't think viral proteins are special. Okay, let's please move on. Thank you, Nancy. So our next speaker is Carrie Parch, and I'm excited to hear how these sorts of structural changes are involved in circadian rhythms. So, okay, can we have your picture, Carrie?
Okay, is this up and running?
Looks good.
Okay, thanks. I want to begin by thanking you for the opportunity to come speak here. In our lab, we're very interested in mechanisms of circadian rhythms, or the way in which life on Earth coordinates their physiology and behavior to run in synchronization with Earth's day-night cycle. And today I'm going to talk about a brief story, a news story that's been the result of a terrific collaboration with Andy Lee Wong's lab at UC Merced and Susan Golden at UC San Diego. And so we're going to talk about the cyanobacterial clock here. And here I'm describing the 3 cyanobacterial clock proteins known as the Kai proteins. And really the central driver of this process is the AAA plus ATPase Kai-C, which is depicted here. It's a hexameric system that has 2 lobed rings here. And many years ago now, Takao Kondo's group showed that, oh, I'm sorry, I lost my pointer. Showed that you could reconstitute circadian rhythms in vitro with these 3 purified proteins and simply the presence of ATP and magnesium as an energy source. And giving rise to this, you see this beautiful rhythm of phosphorylation that shows basically the rhythms of Kai-C phosphorylation over a 24-hour period. And this begins first with the phosphorylation of 1 residue on Kai-C and then a second. This is stimulated during the day by Kai-C and then inhibited by night by KaiB, which sequesters Kai-A so that Kai-C can auto-dephosphorylate. And so you see this beautiful internal timekeeping rhythm of about 24 hours that happens strictly with these 3 proteins in vitro. Now, just by looking at this SDS page gel, hopefully you can sort of see the pain that it takes to follow this process over a 24-hour period, you know, diligent sampling and running the samples. So this is where I'd like to describe a new assay that Andy Lee Wong's lab pioneered that's really opened, I think, the possibilities of studying the mechanism of this clock in really fantastic detail. And this really hinges upon use of a fluorescence anisotropy assay where we can attach a fluorophore to this protein KaiB. And as it undergoes rhythmic association with Kai-C every night, and then it dissociates during the day, you can now see that we can watch this beautiful rhythm happen and unfold before our eyes in real time with very little hands-on activity, right? And so this has really opened the door to asking many key questions about the factors that drive the slow biochemistry on a 24-hour timescale in this system. And so this is where full switching enters the system, or enters the stage rather, and it really hinges on Andy Lee Wong's amazing discovery several years ago that in fact, even though you look at KaiB as a ground state tetramer, any way in which you characterize it spectroscopically, crystallographically, it really looks like a dimer or a tetramer. He discovered that it makes a rare transition to a fold switch monomer. And so in this current configuration I'm showing, it might not be incredibly easy to see how these structures are different. So I'll put them here and color them similarly. So you can see that in fact, the N terminus of both the ground state and the fold states conformation are identical. And it's really about halfway through the protein where you can see that what in fact was a helix becomes a strand and vice versa. This completely undergoes a fold switch. So what I've highlighted here with these 2 spheres are the C-alpha carbons of 2 residues that form the core of the ground state structure, but are here on the sort of surface of the protein and fold switch. And using a bunch of just basic biochemistry knowledge, Andy's lab discovered point mutations that would stabilize this fold switched active form. And so with that, we were able to solve the first crystal structure of KaiB bound to the hexamer of KaiC. And so here I'm depicting that sort of, it's a little dizzying, the KaiC hexamer bound here. And the critical thing that I want to point out, I've colored alternate monomers of this fold switched protein that's docked onto KaiC in different colors here. And the job of this protein really is to, in fact, bind to Ka at night and sequester it. And so I'm not going to go into detail here. This is covered in this paper from 2017. But you can see that one monomer of KaiB is actively sequestering Ka, allowing KaiC to sort of reset for the morning. So we were really struck by data that came out of Albert Hecht's group several years ago that showed that KaiB associates in a highly cooperative manner with KaiC. So these are native mass spec data, and what Yu Snyder showed was that in fact only one molecule or 6 of KaiB would be found to the KaiC hexamer. And we thought this was interesting because this fold switch state is incredibly rare in bulk solution. And so there would be probably a thermodynamic driving force to have a cooperative mode of association to capture this ring. Now I want to point out here that really only one monomer is needed to sequester this dimer of Ka. And so that led us to question how the system might actually come together. And so using the fluorescence polarization based assay, we started to explore the consequences of limiting KaiB concentration and in fact being able to make this repressive complex. So here I'm showing data from the oscillator assay with twice or a 2-fold excess of KaiB relative to Ka, or the sort of standard 1x condition where we have stoichiometric amounts of KaiB present with KaiC. In other words, just sufficient to make this hexamer. And you can see that here in purple, you have beautiful, robust oscillation with approximately a circadian period. But really this goes off the rails as you reduce the concentration of KaiB. When you limit stoichiometry to just half of that, that is to say 3 KaiB monomers per every KaiC hexamer, you see that you can get several low amplitude oscillations, but really this falls apart. And below this, there is no circadian oscillation. So again, I just want to highlight the incredible role of this FP assay in being able to really quantitatively pursue these questions. So this led us to a model where we wanted to explore how KaiB does associate. And so we took the fluorescently labeled KaiB, which I'm highlighting here to remind you that it exists as a ground state tetramer. And then when it associates with KaiC, it takes on this ground state or this active state monomer form. So using 50 nanomolar of this labeled probe, we performed a simple equilibrium binding assay to look at association of KaiB with KaiC. And perhaps not surprisingly, we see this sort of beautiful one-site binding with an approximate KD of about 1 micromolar. We take this to be representative of a distributed mode of KaiB binding, where 1 molecule of KaiB is bound per hexamer, demonstrating the affinity for that initial recruitment step. We then wondered, what if we could seed into this in vitro reaction, just a little bit of that fold switch form to start off cooperative binding and recruitment of this incredibly rare fold switch state. So we did that in the lab by adding very small amounts of a fold switch locked protein that's invisible to this NMR assay, this FP assay, and adding in 100 nanomolar, you see elicited an incredible increase in the cooperativity of association of the labeled KaiB. In other words, planting that flag of the fold switch protein there helped to dramatically recruit the remaining subunits. So if we take a look at this cooperativity interface, the intersubunit interface between KaiB on the hexamer, we can hone in on one particular residue, R22. And in fact, this residue was already shown before many years ago to impact the apparent affinity of KaiB for KaiC, even though in our studies several years ago, we noticed that it wasn't directly at the KaiC interface, which strongly noted a role for cooperativity. So here I'm showing data where we've taken this R22A mutant and put that into our seed stock where we're seeding cooperativity. And I apologize for not having both side by side, but it dramatically reduces cooperative recruitment of subsequent wild-type KaiB, suggesting that intersubunit interfaces between adjacent KaiB monomers is an important step in cooperative recruitment. And so I want to set the stage by going to the next step, which this is the exciting part that really blew my mind when we got to this science. I've told you now that fold switch protein exists in an equilibrium between ground state and fold switch KaiB. But this fold switch form, which is a thioredoxin-like domain, actually has a high degree of structural similarity to the N-terminal domain of an output protein from the clock. And so I haven't really discussed output, but everybody probably understands that if you have a clock that can't communicate that information to the cell, it's not of very much use. And so SAS-A was originally identified as a protein that would communicate, enhance the amplitude and robustness of the clock and communicate information to the cell to coordinate transcription. And so I'm going to post this screenshot of Andy Lee Wong's NMR structure of SAS-A from 2004, where they were puzzling over why, given this high degree of sequence similarity that these proteins have, why didn't they take on the same structure? So I just think this picks up on common themes that both Eileen and Nancy have mentioned, that even when you go looking for it, sometimes you still can't find it, right? Fold switch proteins are tricky in that way. And again, eventually in 2015, Andy's lab showed that, in fact, the fold switch protein has a high degree of similarity to the SAS-A. So this brings me to really the questions that we wanted to ask. So I'm showing here a high resolution crystal structure of just the isolated C1 monomer bound to one KaiB subunit. So this is just sort of taking this out of the hexamer. So to address whether SAS-A could in fact bind at the same site of Kai-C, we solved a crystal structure. And you can see here that binding is centered around this so-called B-loop interface. So we solved a crystal structure of the thioredoxin-like domain of SAS-A bound to the C1 and showed that it bound to C1 at the B-loop in the same rough orientation as KaiB. And this really led us to ponder about sort of nature of interaction here. It had long been posed that this interaction would be mutually competitive, that because they bind at the same site, they must compete for binding. But given this spin that we've put on KaiB association, we wondered whether the thioredoxin domain of SAS-A could in fact seed cooperative binding. So here I'm showing the same type of, we call them 2D titrations because we're looking at binding of KaiB to Kai-C and then looking at the effect of this seeding, cooperative seeding additive here. And you can see that even low nanomolar amounts of that SAS-A thioredoxin domain help to dramatically recruit KaiB to the Kai-C hexamer. And in a whirlwind tour of in vivo data, we made a model of what a mixed hexamer might look like to explore interfaces on adjacent sides, either the clockwise or counterclockwise interface between SAS-A, shown in purple, and KaiB, shown in orange. And suffice to say that mutations of either Q94 or H28 had a modest effect on reducing cooperativity, but eliminating both with alanine mutations had a really significant effect in vitro in this cooperativity assay. And then what was really beautiful to me was that Susan Golden's lab CRISPR'd these mutations into Synechococcus elongatus, and very strikingly, the double mutant of SAS-A that blocks cooperativity from both interfaces, the sort of clockwise and counterclockwise interface, has a dramatic effect on the robustness of circadian rhythms in vivo, reducing the amplitude of them and extending the period. So we wanted to turn back in our last piece of data to this oscillator assay to explore when is it that this cooperativity might be important for circadian rhythms. So we're going to revisit this 0.5x oscillator. That is to say, when KaiB is limiting to create that repressive complex to help sequester Kai-A at night. And as I've shown you before, we can get several oscillations that are of very low amplitude before this dies out. But here you can see the remarkable effect of adding in even just 100 nanomolar of SAS-A, that through the enhancement of cooperative binding, we dramatically restore robustness of the oscillator. So this is typically measured by, or can be measured quantitatively by looking at the damping constant, that is to say the rate at which the oscillation dampens out. And without SAS-A, this is a really poor oscillator, but you can see here that by adding SAS-A, we essentially take this oscillator to almost perfect robustness, as evidenced here by these beautiful oscillations that go on and on and on. And I just want to make a point here that, you know, maybe this is racy. I just want to say I think this could be catalytic in nature, because even with 100 nanomolar of this additive, we're essentially seeing full rescue at only 3% molar equivalent in this reaction. So here we're seeing the side of of cooperativity enhancement, but because SAS-A does compete for binding to the same exact site, we found that if we increase the concentration of SAS-A equivalent to that of KaiC, we actually can effectively compete out all of KaiB. And so there seems to be this nice edge between cooperativity and competition where SAS-A plays a role in tuning the robustness of the clock. And I just want to finish here by pointing out one particular note that there's been a lot of really beautiful quantitative work in this field, and the in vivo concentration of SAS-A relative to KaiB and KaiC comes right at this sort of intrinsic set point of ultrasensitivity between cooperativity and competition. So to wrap up, I've told you that there's sort of concert of protein interactions that happen over the day and the clock. KaiB is cooperatively recruited to KaiC at night, and where it sequesters Kai-A. The SAS-A protein docks onto the same interface predominantly at dusk, and we've shown here that it actually works to cooperatively enhance KaiB binding. It also works to communicate information out to the rest of the cell in the environment, and this is counteracted by another histidine kinase. I don't have time to go into it now, but what's really striking to me again is that both KIK-A and Kai-A here are recruited to the fold switch complex at night, and in fact, they compete for one another. So KaiB, this fold switch protein, truly is a nexus of cooperativity and competition in the clock. So with that, I'd like to thank everyone that did the work. This, again, was a collaboration between 3 labs, and Jeff Swan in my lab did all of the biochemistry and structural work. Joel Heisler in Andy's lab set up this beautiful FP assay that gave us a lot of insight into the clock, and both Cheatham Sanjar and Dustin Ernst in Susan Golden's lab did the in vivo work. And thank you so much for your attention.
Thank you, Carrie. That is a fascinating story. I get so excited when proteins do fascinating things. We have a few questions. We don't have very much time, but I'd like to ask 1 or 2 of them. One interesting question that came up is, are there any similar types of proteins present in humans?
So this is where cyanobacterial clock stands out alone, as far as we know, in terms of the clock mechanisms from all other living organisms. So every other system that we know of works with a sort of different set of proteins. So cyanobacteria is really the one gift that we have in this regard. Yeah,
okay, and you should answer the rest of these questions offline, but one question that I find particularly interesting is this question: does the B loop have a particularly high mobility? And is there some generalization that can be made about the mobility at protein interfaces?
That's a great question. We've never done a molecular dynamics simulation of this, but we do note that it does change conformation between the apo form of KaiC when it's bound to KaiB and when it's bound to SAS-A. So there's definitely movement. One other story I didn't have time to get into here is that actually binding of proteins at that B loop is dependent upon ATP hydrolysis by that domain of KaiC, which triggers a whole set of conformational changes that ripple through the domain down to the B loop. So I think there's a lot more to learn here about cooperativity and conformational change.
All right, thank you very much, Carrie. We're going to move on because we're running a little bit late. And we're going to move on to Jeff Watson, who's going to be telling us about a whole lot of work that was done by an army of undergraduates. So Jeff, can we pull yours up?
Yeah.
And Carrie, please answer all those questions in the box.
All right. How's that looking?
Looks good to me.
All right. So thank you, Eileen, for the opportunity to talk today. As Eileen said, this was, we're a primarily undergraduate institution. So this work was performed in large part by, as she said, an army of undergraduates. So our research in my lab is focused on the enzyme HMG-CoA reductase. And this catalyzes the reaction shown here, which is a reversible 4-electron reduction of this thioester HMG-CoA to this primary alcohol metalonate. And depending on the species from which you take this organism, It uses either NADP, NADPH, or NAD, NADH as the hydride carrier. So in most organisms that have this enzyme, and what most people are familiar with if they know this enzyme, is that it's the rate limiting step in the mevalonate pathway, which is shown here. This is a reductive biosynthetic pathway, HMG-CoA reductase catalyzing this reaction here, that leads to the biosynthesis of isopentenyl diphosphate, or IPP. And then downstream of this, through various other pathways, you get isoprenoid compounds, such as isoprenyl groups for protein post-translational modification, cholesterol, a whole range of molecules that are important in all kingdoms of life. Now, the interesting thing, and the enzyme we study, comes from the bacterial opportunistic lung pathogen, Burkholderia cenocepacia. And we showed a few years ago that this bacterium expresses HMG-CoA reductase, or HMGR, but looking at the genome, you see that it lacks the other enzymes of the mevalonate pathway. And in fact, it uses the non-mevalonate pathway for IPP biosynthesis, which doesn't require the presence of HMG-CoA reductase at all. So it led us to wonder just why does this organism even have this enzyme? And we're still trying to figure out what the physiological role of the enzyme in the bacterium is. So when we started studying it, we noticed some unusual things. So one, BC-HMGR strongly favors the use of NAD, NADH over NADP or NADPH, which suggests it functions in an oxidative direction. Since reductive biosynthetic enzymes typically use NADP, NADPH, oxidative enzymes typically use NAD or NADH. But the more interesting thing that we discovered was that it exhibits unusual kinetics with respect to the substrates of the oxidative reaction in the oxidative direction. So here's an example of something that Eileen mentioned in her opening about double hyperbolic kinetics. So you see that a simple V versus S goes up and then goes up a second time, okay? And I'm only showing varying CoA here, but we also see this when we vary either of the other two oxidative substrates. The other thing that we've noticed is that as you change the enzyme concentration, the Kcat, the rate constant for the turnover step for the formation of product, actually changes as a function of enzyme concentration. You can see that it goes up as you start increasing the enzyme concentration and then decreases dramatically at higher enzyme concentrations. And so this type of behavior, as Eileen suggested earlier on, is suggestive of a morpheein protein. So just to review very quickly, these are multiple non-additive oligomeric states in equilibrium with one another. But in order to transition between these states, for example, a hexamer and a nonamer, they must dissociate to a lower order state, okay? Maybe a monomer, maybe a trimer, and then a tertiary structure change in that lower order state switches to a form that can take on the alternate oligomeric state. So we set out to try and determine whether BC-HMGR has had other characteristics of a morpheein protein. And these were the things that we were particularly interested in looking at. So multiple oligomeric states with different levels of enzymatic activity, structural and functional changes in response to changing environmental conditions, such as enzyme concentration, pH, substrate concentration, and whether BC-HMGR had any other functions. So we are able to show that BC-HMGR is an ensemble of multiple oligomeric states, sensitive to changes in enzyme concentration, shown here on the left, and pH, shown here on the right. And we'll talk about the pH graph here first. So at pH 7.5, we have the blue curve shown here. And this is a size exclusion chromatography chromatogram. And we see three species, actually. We see a hexamer, something that elutes as a nonamer. We're not entirely sure that that's what it is, but that's what I'm going to call it from here on out. And something that's beyond the resolution of our column that we call the bigmer, because we're not entirely sure what the size actually is. And we go to pH 8.3 in red, and you can see that the distribution changes. The amount of nonomer decreases, the amount of hexamer increases, while the amount of bigmer stays the same. As we go to pH 9, we see this chromatogram here in green, where we have almost entirely bigmer with just a little bit of nonomer and not much hexamer at all. And that's the same curve that we see here in red, which is the enzyme at pH 9 and 6 milligrams per milliliter of protein. As we decrease the concentration to a half mg per mil, we see the curve in blue, okay, normalized for the changes in concentration. But it's the proportion of nonomer to bigmer that is really the important thing here. And you can see that there's significantly more nonomer relative to bigmer as we decrease the concentration. And the kinetics results that we see as a function of pH mirror this change in distribution of states. So in green is the pH 7.5 curve, and that's the same curve that we saw earlier. We see the double hyperbolic behavior. As we go to pH 8.3, we maintain that double hyperbolic behavior, but now we see a significantly increased Vmax, okay? And we believe that this is due to the presence of increased amounts of hexamer, which may suggest that the hexamer is a higher activity state than the nonomer. Now, as we go to pH 9, which we presume is predominantly one state, you can see that we have a nearly single hyperbolic curve here. So one of the things that this brought up is just what is the bigmer, and is it relevant to enzyme function? If you were at the webinar yesterday talking about filamentation, you'll know already that assembly of large particles can be pretty significant drivers of enzyme function. So at lower enzyme concentrations, and if we add substrate here, you can see just a little bit of an increase in the optical density at 800 nanometers. So really all we're doing is a quick and dirty check to see if there are large enough particles in solution to scatter light. Okay, so this is optical density, the same thing you do when you're checking the growth progress of bacteria in culture. However, at larger enzyme concentrations, the same amount of substrate added results in a dramatic increase in optical density at 800 nanometers. And you can actually see in the cuvette that the solution has turned cloudy. And we were curious whether this was just a non-specific aggregation and precipitation issue. So we looked to see if we could reverse it. And we are able to reverse that assembly process. So we add NAD+ to assemble the particle. We add NADH, and that results in a decrease in the large particle. Add more NAD+ to get it to assemble again. Addition of CoA, as you can see here and here, both result in a decrease in the prevalence of this bigmer. And as we showed in the pH 9 kinetics data, that bigmer remains active, whether it's disassembling to the nonomer and catalyzing the reaction or catalyzing it as the large particle, we're not sure yet, but it certainly is a relevant species. So just to answer the first two questions, yes, BCH-MGR does have characteristics of a morpheein protein with multiple oligomeric states, changing in response to things like enzyme concentration, pH, and substrate concentration. So what I want to talk about right now is whether or not BCH-MGR has other functions. And so we were able to determine the crystal structure of BCH-MGR, and we got a surprise. We co-crystallized with coenzyme A, but we also found ADP bound to the enzyme as well. And this wasn't a huge surprise where it bound, since this is exactly where the ADP moiety of NAD+ would bind, but no HMGR has ever been shown to use ATP or ADP. And the fact that we didn't add it suggests that it co-purified, possibly, which suggests that it binds with relatively high affinity. I'll note that the crystal structure we were able to determine is of the hexameric form. We haven't been able to get structures of the other forms yet, but we're working on that. So what does ATP or ADP do to this enzyme? We're able to show that BC-HMGR can hydrolyze both ATP and GTP, data I'm not showing here, but we weren't sure what purpose it was for. As I said, no HMGR to date has ever been shown to have ATPase or GTPase activity. So one of the things that we tested is adding ATP and GTP to a mixture of the enzyme, mevalonate, and NAD+, which are the two of the three substrates required for the full reaction. And then we looked for the appearance of NADH at 340 nanometers. So as you can see, adding CoA results in an increase in NADH production, as we would expect. That's the normal reaction. Adding GTP is the curve in purple here. It does nothing at all, even though GTP can be hydrolyzed in this situation. ATP, on the other hand, demonstrates a dramatic increase in production of NADH, far more than you get from what's considered the natural substrate, coenzyme A. Also, in a similar fashion to NAD+, shown here in green, adding ATP to BC-HMGR induces a dramatic increase in the bigmer particle, as judged by OD 800. GTP does none of that. However, GTP is capable of disassembling a pre-assembled particle with NAD+ pre-incubated with the enzyme. ATP is able to further assemble that particle. So BC-HMGR may have other functions. ATP and GTP hydrolysis, certainly ATP appears to promote a redox reaction, product unknown thus far, and both nucleotides do affect the assembly and disassembly of the large particle. So with that, I'm going to come to an end. I do want to acknowledge that army of past and present students, particularly the students whose work I showed here today. Riley Peacock, who's just finishing his PhD at UCSD, Chad Hicks and Sophia Dewing, who are in PhD programs at the institutions shown there. Crystallography was done in collaboration with Chulhee Kong's lab at Washington State University and his students Xander Walker and Kevin Lewis. Funding shown here at the right. And thank you for your time, and I'll be happy to answer questions.
And I will unmute and show you my face. And there are a few questions. For example, Emily Arturo asks, is there any evidence to suggest that any mammalian HMGR has these multimeric properties?
So there are some interesting oligomeric state differences between different types of HMGRs. So the mammalian ones are all, to our knowledge, are all tetramers, dimers of dimers. Reductive bacterial HMGRs appear to be only dimers, and the presumably oxidative bacterial HMGRs seem to be either dimers or hexamers or both. No other HMGR has been shown to form these other oligomeric states.
What you call bigamers.
Bigmers, yeah.
Bigmers. Another question is, if your enzyme is knocked out in the Burkholderia, are the bugs viable?
Boy, I'd sure like to know. That's a project that we're starting this fall. It is a BSL-2 level pathogen, and we are a primarily undergraduate institution, but we're actually able to do that work here on campus now. It is a little bit difficult to do knockouts in BC, but it has been done, and I've got a new army of students who are going to start working on that project.
Okay, and now I have another question, which sort of is a question I think many of us have dealt with. So here someone writes, I also came to know with a protein I am working on, bigmer, using your words, at higher pH. Then I thought it was just an aggregate due to high pH. Am I wrong?
you may
I
or may
love that question.
Yeah. It's so at pH 9, we're able to concentrate our protein to nearly 40 milligrams per milliliter concentration. It loves that bigmer, but it does come back apart. And what I would suggest really is try adding substrates. Try doing those other things, changing the enzyme concentration, et cetera, to see if you can get it to come back.
Right. I mean, it turns out for the PBGS system, you need turnover to be occurring to get this octamer to hectamer transition. Which is a whole other story. All right, there are a couple of other questions. I hope you'll answer them offline. And let us please move on to Constance Jeffrey, who's going to talk to us about her favorite topic, which is moonlighting. And here we're going to hopefully learn about moonlighting and alternate structures. So Connie, can you bring your slides up?
Okay, how's that? Okay?
Yes, I see your slides and your picture. Go for it.
Okay, great. First, I want to thank Eileen for putting together this very interesting symposium and for giving me a chance to tell you all a little bit about my lab's work with moonlighting proteins. My focus will be on moonlighting proteins where changing shape promotes changing function. But first, I'll introduce the idea of moonlighting proteins. So we're defining moonlighting proteins as multifunctional proteins where two or more physiologically relevant biochemical or biophysical functions are performed by one polypeptide chain. And one of the proteins we worked on in my lab with this is phosphoglucosisomerase. This is a cytosolic enzyme in glycolysis and gluconeogenesis that converts glucose 6-phosphate and fructose 6-phosphate. So that's while it's inside the cell. It's also a secreted protein, and when outside, it can interact with cell surface receptors on other cells, and it can do things like promote pre-B cells to differentiate into antibody-secreting cells, and it can interact with other cells for performing other types of functions. Oops. Okay. So one of the things that my lab did was search around for other proteins that have two very different functions like this. Originally, 20 years ago, we thought, well, this was unusual, but we're finding that there are actually hundreds of proteins that have two different functions in one polypeptide chain. So we created the MoonPro database, and this is an online database. It's curated, it's manually curated. So these are proteins that we've found in the literature with experimental evidence for each of the two different functions. And this is not proteins that just have suggested additional functions from being found in unusual locations or other information like that. We're looking for proteins that definitely have two different demonstrated functions. So on a database, you can search using the amino acid sequence or various names of the protein, and it'll bring you to a page like this where we have various names for the protein, sequence information, structural information, Go terms, and a clear description of the two or more different functions for that protein with additional information, including references where they showed that the protein has each of those functions. So in our database so far, we have over 350 moonlighting proteins, and they're very diverse. They're found throughout all the kingdoms of life, and they include a lot of different types of proteins. So there are enzymes that have a second function as a chaperone or as a secreted cytokine, like that phosphoglucosisomerase example. Some enzymes are also transcription or translation factors, or they might be part of the cytoskeleton, for example. There are also some moonlighting proteins that don't have catalytic activity, but they still have two different functions. So, for example, some ribosomal proteins also are transcription or translation regulators. There are also a lot of different ways for a protein to switch between functions. So if we think about that cytosolic enzyme like phosphoglucoside inside the cell, it might have a second function outside the cell when secreted, such as a growth factor. Some proteins switch function when they're expressed in different cell types with different macromolecules and other cellular components. In some cases, binding of a small molecule, substrate product, other ligand, can cause a switch between functions. For example, enzyme to translation or transcription factor. Some proteins will have one function as a monomer or homomultimer, but will switch functions when they interact with another protein. And some will have a function again as a monomer or homomultimer, but will join in a multi-protein complex such as the proteasome or possibly the ribosome to perform another function. So we have a great deal of diversity in terms of functions and how they can switch between functions. And for this talk, I wanted to think about what is happening with the structure of a protein when the function changes. And I'm going to summarize on the next few slides the wide range of very small to very big changes that we actually see in some of these moonlighting proteins. So first of all, I'm going to talk about some proteins where we don't really think there's much of a three-dimensional change in the proteins as they perform their different functions. And an example of this are the taxon-specific crystallins. These are proteins that are often ubiquitous enzymes found in many species. So, for example, arginose-succinate lyase catalyzes a reaction in many species. But in some species, they're expressed at a very high level in the lens of the eye, where they have more of a structural role. And as far as we can tell, that structure of the protein doesn't really change much between these two functions. It would just have the appropriate physicochemical properties to perform this other role. And so it was adopted during evolution in different lineages to perform this function in the eye. We also see proteins that have a little bit more of a change in shape, but still not an overall change in the tertiary or quaternary structure. And an example of this is fructose 1,6-bisphosphate aldolase phosphatase from some hypothermophiles. This enzyme catalyzes two consecutive activities in gluconeogenesis. And the main change as it does these two different things are just some loops in the active site pocket. So both of these catalytic activities happen in the same pocket. And we have several loops. For example, this blue segment here that changes here, and then a big change right there. And there's a red loop. See how it also changes shape. And there's a little bit of a smaller yellow loop down here. These loop changes bring in different catalytic amino acids at different times so that you can have different arrangements in the active site. And it also affects binding of metal ions that play a role in catalysis. So in this protein, again, the overall tertiary or quaternary structure doesn't change, just localized regions in the active site. Now, there are some proteins that have a somewhat bigger change, and aconitase is an example of a protein where you have some significant conformational changes within a subunit. Aconitase is an enzyme in the citric acid cycle, converts citrate to isocitrate. To do that, it uses an iron-sulfur cluster. But when cellular iron levels are low, it loses that iron-sulfur cluster, and it undergoes a very large conformational change. So there's a huge movement of domains relative to each other, exposing a new surface that RNA can bind to. So the enzyme becomes a translation regulator that promotes the expression of proteins that are involved in bringing more iron into the cell. We can have even bigger changes in the structure of a subunit when we consider some of the intrinsically disordered proteins that are moonlighting proteins. So the intrinsically disordered proteins don't have a single tertiary structure in the cytoplasm. They're very, very flexible, considered unfolded, but some of them will actually fold, or part of it will fold when it interacts with a protein. And in some cases, this folded part is different when it interacts with different proteins and performs different functions. Okay, so we can have this intrinsically disordered to folded into sometimes different folds with different partners to perform different functions. We also have a few proteins that are called metamorphic proteins that have two stable tertiary folded. So both of these forms are folded. This is not unfolded to folded, but two folds that can interconvert and have different functions. Or some metamorphic proteins have different functions with the different folds. An example of that is lymphotactin that has LTN40 has mainly beta sheet structure, whereas LTN10 has partial beta sheet plus some alpha helical structure. The LTN10 form of the protein is an agonist for a G protein coupled chemokine receptor, but the LTN40 protein binds to heparin and not the receptor. And finally, we come to the morpheeins, which change in conformation of a subunit, and they also change in their quaternary structure as well. So a few of these have been mentioned already. The VP40 protein has 3 structures with 3 different functions. Ribonucleotide reductase has heterotetramer and hexamer forms, and it's also an inhibitor of another protein involved in cellular proliferation. Now, porphobilinogen synthase is an interesting example of the prototypical morpheein that undergoes changes in tertiary and quaternary structure as a type of regulation. But it's also a moonlighting protein because in addition to this, it has a second function as a proteasome inhibitor. So in summary, we have a large number of proteins that are found to have 2 or more different functions. This change between 2 functions can be achieved by several methods, and structural changes as they switch from one function to another can be very diverse. Everything from what we think is a tiny or insignificant change in structure through conformational changes of a subunit, including small movements of loops or larger domain movements. And then we can have changes in the tertiary fold, intrinsically disordered to folded or complete refolding of domains. And then we can have both changes in quaternary structure with conformational change of subunits, which is the morpheeins. So I'd like to thank all the various students through the years who have helped put together the database, construct the database, as well as do a lot of the annotation of the database, and a lot of the funding agents who have helped us, supported us as we've studied moonlighting proteins through the years. And also welcome any suggestions. A lot of these proteins were found through serendipity, and they kind of pop up throughout the literature in all different interesting places. So feel free to send me more suggestions of proteins for adding to our database. We're actually, as we're social distancing, we're actually providing opportunities for a lot of undergrads to have research projects and helping with the annotation, and also in analyzing sequences and structures. So we're actually doing a lot of annotating over the summer. So, and thank you once again for all your time and attention, and for this opportunity to tell you about our moonlighting proteins. So I'll do that.
Okay. Thank you so much, Connie. I have a question.
Sure.
So there's a protein, glyceraldehyde 3-phosphate dehydrogenase.
Yes.
Everybody uses it as a standard, you know, on their gels, but it has, if I'm not mistaken, 7 different moonlighting functions.
At least.
At least. So is there any information out there about whether or not that protein samples structures other than the quaternary crystal structure that's been in the literature for quite a while?
I haven't looked at that one specifically, but I think it's something that's definitely worth looking into for a lot of these proteins. I think it would be an interesting question of how we can, I think it'd be interesting to have a systematic way of looking at a lot of these proteins and seeing if we could promote another structure and perhaps trap that for crystallography or cryo-EM or something. So I don't know of one specifically off the top of my head for GAPDH, but I'm definitely interested in seeing if we can do additional structural studies with some of these proteins and find additional structures.
Yeah, it's a fascinating possibility, particularly since what's in the PDB are the forms of the protein that form a crystal.
Yeah, yeah.
Okay, we have a few other questions. Let me make sure I get them all. Are proteins that have different functions in monomer versus self-assembled considered moonlighting proteins, that is prion-like proteins? Would they be included in the MoonProt database?
Okay, yeah, that's a very good question. So there are prion proteins that have multiple functions that are physiologically relevant. So we, I think there's some yeast proteins, if I remember correctly, that do different things when soluble and in the prion. So those we would consider moonlighting. We tend not to include proteins where the extra structure is not sort of healthy related. So some proteins will gain a function due to mutations or in disease. So there are some that form prion or amyloid type structures. We haven't necessarily included them in our database, and that's something where it's kind of a question of, you know, we think of it as not a normal function of the protein, I guess is we're trying to think of things that would be the usual function of the protein. So in some cases, like in yeast, it is an actual part of the normal function of the protein. So then it would be included. Okay.
Yeah, there's another one. I lost it. So this is an interesting. Can we use existing information on moonlighting proteins and their structure-function relationships to design therapeutic proteins? Has anyone done it previously?
So one of the reasons I'm interested in moonlighting proteins and cataloging them is to think of them as, what does it tell us about protein structure and function, of course, and predicting structure and function, but how can we also use this information to perhaps design other proteins with different functions? So some of these proteins that have multiple functions have different binding sites, maybe even different active sites. So how did these additional sites develop in nature after we already had a protein doing one thing, right? So we had a scaffold protein. It has one function, maybe one active site. Now it's doing something else. So how was that scaffold used to help form another binding site, another active site? And can we use that in design? So perhaps as therapeutics or manufacturing. And in some cases, therapeutics can be very useful if they do have multiple abilities. So you might want to have for example, a design protein that can bind to something, maybe carry it to a particular place in the body, and then release it. So some of these ideas of how do proteins do different things and change conformations as they do them could inform future development of proteins as therapeutics or for other purposes as well.
Okay, and the last question here is, can we find multiple conformations of a metamorphic protein in the same oligomeric state? And this is something that we're working on right now, in that we're working on phenylalanine hydroxylase that has two structurally very distinct tetramers. And that makes it a little more difficult experimentally to discriminate between them because they're both tetramers. But what do you have to say on this?
Yeah, so I guess I'd have to, I don't know if I can really add to that. I think that's a very interesting example right there. So there are some that the same oligomeric state, but yeah, I can see how usually we would separate those by something like gel filtration or other chromatographic methods, and that would be very hard. So I would have to say, other than your suggestion, I'm not sure if I know of any other specific examples.
Well, one thing that I didn't stress when I introduced this whole topic a couple of hours ago is the fact that these different oligomers can often be separated by ion exchange chromatography. Okay. Which is how we learned about the PBGS octamer and hexamer. They're chemically the same, but they're structurally distinct. They have different surface charge, and they will separate on an ion exchange column. And we have used that extensively in our phenylalanine hydroxylase.
Yeah.
Okay, thank you, Connie.